Welcome to Human Physiology. Today, we are diving into the life-sustaining systems of oxygen and carbon dioxide transport in the blood. Imagine your body as a bustling city with millions of homes. We are going to look at the highways and delivery trucks that keep every single cell alive and functioning. Let us start by understanding how oxygen travels. Think of your cells as homes needing fresh groceries, which are the oxygen molecules. The blood is the highway, but oxygen cannot just float freely in large amounts because it is nonpolar and does not dissolve well in water. In fact, only about 1.5% dissolves directly in the blood plasma. To deliver the massive amounts we need, the body uses specialized delivery trucks called red blood cells. Inside these cells, oxygen binds to hemoglobin, which carries the remaining 98.5% of our oxygen supply. Now that we know hemoglobin is the primary carrier, let us look at why its function is so critical. Hemoglobin contains iron-rich heme groups that bind to oxygen reversibly. This reversibility is essential because hemoglobin must grab oxygen in the lungs and easily let it go in the tissues. Understanding this helps us diagnose medical emergencies. For instance, in anemia, a low red blood cell count means there are not enough trucks to carry oxygen. In carbon monoxide poisoning, toxic gas occupies the seats on hemoglobin, blocking oxygen entirely. Without hemoglobin, our hearts would need to pump over 100 liters of blood per minute just to keep us alive. This brings us to a fundamental tool in respiratory physiology, the arterial oxygen content equation. Conceptually, this formula simply adds the large amount of oxygen bound to hemoglobin to the tiny amount dissolved in the plasma. Let us break down the symbols. CaO2 represents the total arterial oxygen content in milliliters per deciliter. Hb is the hemoglobin concentration. The constant 1.34 is the oxygen carried by 1 gram of saturated hemoglobin. SaO2 is the oxygen saturation percentage, written as a decimal. Finally, 0.003 is the solubility coefficient, multiplied by PaO2, the partial pressure of oxygen. If hemoglobin increases, total oxygen rises massively, while changes in partial pressure have a much smaller effect. Before we move on, let us see this with a concrete example. We want to calculate the total oxygen content for a healthy adult. We are given a hemoglobin concentration of 15 grams per deciliter, an oxygen saturation of 98%, and a partial pressure of 100 millimeters of mercury. We chose this formula to account for every single molecule of oxygen. First, we calculate the bound oxygen by multiplying 1.34 by 15 and then by 0.98, giving us 19.7 milliliters. Next, we find the dissolved oxygen by multiplying 0.003 by 100, which yields just 0.3 milliliters. Adding them together, we get a final answer of 20.0 milliliters of oxygen per deciliter. This proves mathematically that almost all oxygen is carried by hemoglobin. Now that we have calculated oxygen content, let us visualize how hemoglobin behaves using the oxygen-hemoglobin dissociation curve. Looking at this graph, the horizontal axis represents the partial pressure of oxygen in millimeters of mercury, while the vertical axis shows the percentage of hemoglobin saturation. Notice the distinct S shape of the curve. The steep region on the left shows where oxygen is rapidly loaded or unloaded, which corresponds to the environment in our tissues, marked here as venous blood. As we move to the right, the curve flattens into a plateau region. This represents the arterial blood in the lungs, where hemoglobin becomes almost fully saturated with oxygen. This diagram beautifully teaches us how hemoglobin is perfectly adapted to grab oxygen in the lungs and release it exactly where the body needs it most. Having explored oxygen delivery, we must now look at the reverse journey. Just as a city needs garbage collection, our bodies must remove waste. Carbon dioxide is the primary metabolic waste product generated when our cells produce energy. Why does this matter? If carbon dioxide is allowed to pile up, it turns the blood acidic, creating a highly toxic environment that can completely shut down cellular functions. Therefore, the blood must efficiently transport this carbon dioxide away from the tissues and back to the lungs to be exhaled. This reverse journey is just as critical as oxygen delivery for keeping the body's internal environment stable and safe. So, how exactly does the blood carry this waste? Carbon dioxide is transported through three distinct mechanisms. First, about 7% is simply dissolved in the blood plasma. This is higher than dissolved oxygen because carbon dioxide is more water-soluble. Second, about 23% binds to the amino groups of proteins, primarily hemoglobin, forming a compound called carbaminohemoglobin. Finally, the vast majority, about 70%, is converted into bicarbonate ions inside the red blood cells. This massive conversion does not happen on its own. It is rapidly catalyzed by a specialized enzyme called carbonic anhydrase. Let us look at the mathematical and chemical representation of this crucial conversion. Conceptually, this formula shows how a volatile gas is safely transported as a dissolved salt. Carbon dioxide, or CO2, combines with water to form H2CO3, which is carbonic acid. Because carbonic acid is unstable, it immediately dissociates into a hydrogen ion and HCO3-, which is the bicarbonate ion. The double arrows mean this reaction is reversible. In the tissues, where carbon dioxide is high, the reaction shifts right to create bicarbonate. In the lungs, where carbon dioxide is exhaled, the reaction shifts left, turning bicarbonate back into gas. This is the cornerstone of acid-base balance in the human body. This chemical reaction we just saw leads us to an important biological realization. The conversion of carbon dioxide into bicarbonate is a stroke of biological genius. Why? Because not only does it provide a highly efficient way to transport massive amounts of waste gas, but the resulting bicarbonate ions also act as the body's primary pH buffer system. This means the body literally repurposes its own waste product to protect the blood from becoming too acidic or too alkaline. If this transport system fails, such as in severe respiratory diseases, carbon dioxide accumulates. This leads to a dangerous condition called respiratory acidosis, which can cause coma or even death. While the vast majority of carbon dioxide is carried as bicarbonate, we must not ignore the small fraction that is simply dissolved in the plasma. This dissolved portion is incredibly important because it is the exact fraction that determines the partial pressure of carbon dioxide in the blood. This pressure is the physical force that ultimately drives the diffusion of carbon dioxide from the blood into the lungs so it can be exhaled. To understand exactly how much gas is physically dissolved in the fluid based on its pressure, physiologists rely on a fundamental principle of physics known as Henry's law. This brings us to Henry's law. Conceptually, this law states that the concentration of a dissolved gas is directly proportional to its partial pressure. The formula is beautifully simple. C equals P times alpha. Here, C is the concentration of the dissolved gas, measured in millimoles per liter. P is the partial pressure of the gas, measured in millimeters of mercury. And alpha is the specific solubility coefficient for that gas in the blood. Physically, this relationship shows that if the partial pressure of carbon dioxide increases, the amount of gas dissolved in the liquid will increase proportionally. It is a direct, linear relationship applied to human physiology. Let us apply Henry's law to calculate the dissolved carbon dioxide in venous blood. We are given a partial pressure of 45 millimeters of mercury and a solubility coefficient of 0.03. We use this straightforward multiplication to isolate the specific fraction of carbon dioxide not bound to proteins or converted to bicarbonate. We substitute 45 for P and 0.03 for alpha. Multiplying these together, we arrive at our final answer: 1.35 millimoles of dissolved carbon dioxide per liter of venous blood. This specific dissolved fraction is crucial because it exerts the pressure needed to push the gas out of the blood and into the lungs. We've covered the full topic of gas transport, so let us reinforce the most important ideas. First, red blood cells and hemoglobin are absolutely essential for survival, carrying nearly all the oxygen our tissues need. Second, we saw how the arterial oxygen content equation perfectly combines the large bound fraction with the tiny dissolved fraction. Finally, carbon dioxide transport is a multifaceted process where the body brilliantly converts 70% of its toxic waste into bicarbonate ions, safely transporting it while simultaneously buffering the blood's pH. You now have a solid understanding of how these life-sustaining systems keep us alive every single second. Great job!